Hey Bio Team. So if you guys have ever seen a protein, you'll know it's a really complicated, complex structure. And you've already learned a little bit about what causes that structure. And the base of every protein is something called its primary structure, which is the sequence of amino acids that join it together. So it's a really long strand of amino acids. Some are polar, some are nonpolar, and some are charged, that are joined together by covalent peptide bonds that form during dehydration synthesis. But depending on the primary structure, depending on whether the amino acids are polar, nonpolar, or charged, the protein can start folding and twisting together into what's called secondary structures. And so there's two types of secondary structures. The protein can twist into this thing called an alpha helix, or it can start to crease and fold into something called a beta sheet. And so what holds the secondary structures together, what allows them to twist or fold into beta sheets, are hydrogen bonds. Now these secondary structures, the alpha helixes and beta sheets, they can start to fold too into three-dimensional shapes called tertiary structures. And so you can see here where these beta sheets and alpha helixes have started to kink and fold into three-dimensional structures. And what causes the third dimension, and what causes this tertiary folding, are things like salt bridges between the ionic amino acids, or things called disulfide bonds between amino acids with available sulfur in them. And then last, after a protein has formed a tertiary structure, it might be able to form a quaternary structure as well by interacting with other tertiary structures of different proteins. And as for what holds that together, that can be any of the above types of bonds. So what happens if this carefully constructed shape of a protein changes? Well, if you change the primary structure of a protein, that is, you change the sequence of amino acid, there's the potential for all the other structures to change too. So, for example, we have this protein here. We can see its long strand of amino acids, and you can see that right now the protein is being bent and folded by a salt bridge. You've got a positive amino acid on one side of the strand and a negative amino acid on the other, and that's kind of connecting this protein into a clover leaf, into this kind of loop here. Now, if you were to swap out one amino acid that's charged with, for instance, a nonpolar amino acid, well then all of a sudden the amino acid strand wouldn't be able to hold together. It would stop attracting to each other. The protein could unravel. And this is a problem because proteins depend on their shape to function. The classic example of this is a protein called an enzyme. Enzymes are the things that we use to break apart and put together our nutrients in our body. And the way an enzyme works, you can see this enzyme down here, is that they have a very specific shape that matches a specific substrate, a specific resource in our body. And so the substrate fits into the enzyme where it can get broken down or synthesized onto something else. However, if the enzyme's shape changes, you can see there isn't a lot of room for error on this enzyme shape. If this shape of the enzyme were to change even slightly, then the substrate wouldn't be able to interact with the enzyme as easily, and the enzyme would stop working. And so we call the process of a protein misfolding, or a protein folding in the wrong direction, coming undone, we call this denaturing. And that's it. At this point, you guys have some practice problems. We'll see you next class.